Enzymes and digestion, AQA biology. Key terms: digestion, breaking down of food into smaller molecules. Physical breakdown, protein broken down into smaller pieces. Chemical breakdown, large molecules broken down by hydrolysis. Digestion enzymes, produced by specialized cells, work with different foods as enzymes specific to substrates. Digestion. Now this is a little diagram of the digestion system, and you have the esophagus, stomach, ileum, large intestines, rectum, salivary gland, and the pancreas. Now, examples of chemical breakdown are carbohydrates, lipase, and protease. Now, carbohydrates breaks down carbohydrates to form monosaccharides. Lipids, lipase breaks down lipids to fatty acids and glycerol. And proteases break down proteins into amino acids. Digestion enzymes examples. Well, amylase breaks down starch, catalyzes starch to form maltose. Lipase breaks down lipids to form monoglycerides and fatty acids. Endopeptidases hydrolyzes peptide bonds within a protein, and exopeptidases hydrolyze peptide bonds at the end of the protein molecule. So if you think about it, the way I like to think about it is exo is external. So if it's external, it's at the end. The structure of the ileum, well, it has a folded wall, which increases surface area, as do the villi, and a rich blood supply. Digestion of carbohydrates. Well, food is broken down by amylase. Well, carbohydrates are broken down by amylase and membrane-bound disaccharides. Amylase catalyzes starch forming maltose, and a disaccharide attached to the membrane is broken down into monosaccharides. Monosaccharides are then transported by transporter proteins. And then the absorption of carbohydrates are glucose and galactose moved by co-transport, and fructose is moved by facilitated diffusion. Lipid digestion. Lipid digestion occurs by the fact that lipase is made in the pancreas and bile salts are produced in the liver, and it results in lipids being emulsified, which form droplets which increase the surface area. And once the lipid is broken down into fatty acids and monoglycerides, they stick to the bile salts to form micelles. And micelles move monoglycerides and fatty acids towards the the ileum, which then moves towards the epithelial. And this constantly breaks down and reforms to release the fatty acids for them to be absorbed. The micelles are not absorbed into the epithelial. Now, monoglycerides and fatty acids are lipid soluble, so are able to diffuse across, whereas micelles are not. And the absorption of lipids. Micelles attach to the microvilli, and the digested product diffuses passively. Short and medium chains don't need micelles because they're small enough to diffuse across by themselves. And then they diffuse into the ileum, into the epithelial cells. And this is a little diagram showing what's occurring. So if you want to pause the video here, you can just look at the diagram because that's basically what's being explained already. Protein digestion. Protein digestion occurs as proteins are broken down by different proteases, and enzymes catalyze the conservation of proteins into amino acids via the route of hydrolysis. And it uses endopeptidases, dipeptidases, and exopeptidases. Examples of endopeptidases are trypsin and pepsin. Examples of exopeptidases are dipeptidases. And where are trypsin synthesized? Well, it's synthesized in the pancreas and is secreted in the small intestine. And absorption of amino acids are done by diffusion and co-transport. And this is a little diagram showing what occurs. So you have your exopeptidases, your endopeptidases, and once they're broken off, they're absorbed into the ileum, and then into the epithelial cell. And then once in the epithelial cell, is able to go into the capillary tube. Now that's all there is really for diffusion, for digestion and absorption. And if you like this video, then please like and subscribe. And the next video will be about mass transport, more specifically hemoglobin. Thank you for listening.